microcirculation. It's the structure of microcirculation in the capillary system. So what kind of gets funky with this lesson is towards the end of it. So the whole kind of idea and concept, we'll look at this more in a second, is we need to get stuff into the tissue and pull stuff out of the tissues in order for it to keep working. Like we need to give oxygen, we need to take carbon dioxide. We need to drop off glucose. We need to pick up, like if there's other chemicals, other stuff, lactate, excess of any of the waste products, anything that essentially we're using with those muscles or with any other tissue, we need to be able to move it back and forth. For the most part, we kind of focus on like the muscle type system for microcirculation because that's where it's going to be like the most impacted. Either muscular system or like the intestinal type system or like the kidneys and stuff. Those guys, that's kind of going to be our big focus on like microcirculation. So kind of getting into it, we're going to look at the structure of the capillaries, the function. There's 3 different types of capillaries we want to know. We'll want to know what they are, what they do, and where they are located. The substances changing back and forth, and the factors that contribute to the exchange that occurs in microcirculation. Like I was saying, it's going to be a good amount of gradients. So like there's less oxygen here, there's more proteins this way, so kind of fluid gets pushed and pulled with a lot of like the osmosis type stuff. There's osmotic pressure, and there's it's basically what the name of the other pressure is, but we'll see it when we get to it. In the lymphatic system and the lymphatic flow. So a little bit of the intro here. We have capillaries on this side. We have the arterial, opposite the venule. We have the meta-arterial, which is like the middle. The meta-arterial is the one that's always going to be connecting our arteriole to our venule, so connecting the artery side of things to the vein side of things. We have all of our capillaries. The whole thing, the whole web of capillaries is called the capillary bed. The capillary bed equals all the webbing of capillaries in an area. And all of our capillaries have these little pre-capillary sphincters. That's going to control the capillary flow. So if we're not using that kind of those tissues and we don't need a bunch of blood running into there, like let's say it's leg day, so we're really working out the legs, a lot of the muscles, the capillaries and stuff in the gut or in our upper body muscles, that kind of stuff, the ones that we're not using for that activity, those capillaries, those sphincters are going to close and blood flow is going to stop. Let me actually see if I can find... There's a really cool video. I know that I have it saved. I'm going to see if I can find it really quick because it's one of my favorite videos to show where you can actually watch this in real time happen. Let's see. It should be under the microcirculation PowerPoint of my stuff. Well, that pulls up, but if I can see if I can find it, I'll keep going. So there we go. So the whole purpose of kind of like what we said of this site is to exchange between the tissues and the blood. That's the key word, is exchange. That exchange means we are dropping stuff off from the blood to the tissue and picking stuff up from the tissue to the blood. You can kind of think of this, the example that they'd use is kind of like a garbage truck and delivery truck rolled into one. where we're dropping off the packages that they need and we're picking up the stuff at the same time as well. Let me see if I can find, nope, not that one. No, it's in here. There's a really, here it is. This video is really, really cool. Open it up. So this is actually live stream. Like, see all of these different vessels going off to the side, and just watch the blood's flowing through those. And it should, wait for everything to close down. There it goes. You can watch this vessel right here. It's getting smaller, it's getting smaller, it's getting smaller, more and more narrow, until it literally just disappears. And it's cut off all the blood flow to that area because that blood flow is not needed, and then it's going to start opening again. So you can see that that's exactly what's happening within our tissue. I really like that video because it shows like what's actually happening. Okay, so we deliver the nutrients, the oxygen, the solutes, and all that stuff to produce ATP, and we remove the waste, the metabolic waste, CO2, lactate, any other products. And there's a ton of capillaries in our body. Tons and tons and tons and tons and tons of capillaries. Because they're what go into all of our bits and pieces of tissue to make sure that it has blood. So because there's so many capillaries, we talked about the large cross-sectional area. We talked about last week how if we have a large cross-sectional area, it's related to how fast. We talk about velocity. So velocity is how fast the blood moves through. Large cross-sectional area means that the blood velocity is split between a bunch of different channels, so it's going to move a lot slower. And the arterioles carry all of that blood into that capillary circulation, going all the way through. And also because we split all that kind of stuff, and we're going from arterioles to the vein. Remember, the venous side has a really, really low pressure. And as we get further and further away from the main arteries, our blood pressure continues to drop. So the capillaries, and also because there's such a large cross-sectional area, the blood pressure there is really, really low, which is important because we want things to stay in the capillaries for a while to let the exchange happen. We don't want the blood going too fast through there. This is just some anatomy stuff, looking at the structure itself of the capillary. You'll want to know some of these words. You'll want to know the endothelium. The entire capillary is literally a single layer of cells all around. It's super thin. You have this thing called an intracellular cleft or slit pore, because the whole point of the capillary, is to let things go out. So we need to be able to have, so there's our intracellular cleft right there. We need to have these little openings for stuff to leave the capillaries and to enter the capillaries, and that happens in between the cells, which prevents the plasma proteins, but lets the small solutes through. Just some more about the structure. The caveolae, these little cave-like structures there, they're thought to serve in transcytosis. Transcytosis is the transport of those macromolecules from one side of a cell to the other. So think like cells can kind of go into here and it'll close off and float to the other side, and then do endocytosis to let it through the other side. This isn't super important. I would just remember these two words. Remember endocytosis and remember transcytosis are correlated with caveolae. That's the only way he's going to ask that type of question. It's more like an association type thing. So the different types of capillaries depend on what tissue they're going to and what kind of stuff they need to drop off in that area. So these are our continuous capillaries. These have really tight junctions. They're not leaky. They only allow like the small solutes to cross. So these are continuous. When we look at this, the skin, the nervous tissue, connective tissue, and muscle tissue, all of these kind of see, these are the least leaky. These are the least ones that are going to let stuff out of the capillaries. He has in here also in the brain. So again, that nervous tissue. Fenestrated capillaries. These tend to be the ones that get talked about a lot. These have slightly larger gaps. These allow stuff like ions and peptides to go through, especially like the kidneys, the small intestine, endocrine glands, all of those ones. If we think about the function of kidneys, the small intestines, endocrine glands, the kidneys, their job is to filter stuff out. So they need to have more availability for stuff to come out. And so for them to be able to pull stuff out of the capillaries in order to filter it and put it into the urine. Same with the small intestine. The small intestine needs to be able to push a lot of stuff towards the blood. We got to get the nutrients and everything that we have that we're getting out of the small intestine, the water and stuff as well, from the small intestine, get it into the bloodstream where we're going to need it. So, excuse me. It's mostly found in the glomeruli in the kidneys. You'll talk more about the kidneys. We'll talk about a little bit about the kidneys this trimester. Next trimester in Phys 2, we talk a lot about the kidneys. There's a big structure on the kidneys. So we have continuous, we have fenestrated, and then we have sinusoidal, kind of like a sinus cavity. Sinus cavities are really big cavities. I have a lot of them in our face. We have our maxillary sinus, our frontal sinus, the endothelial sinus, ethmoid sinuses. So these guys have the largest gaps, and these allow the big stuff like proteins and red blood cells to come out of them. So then we think, okay, what kind of things, for what reason do we need to bring blood cells and stuff out? Our body is continuously making new blood cells, and so we also need to break down and recycle old blood cells. These types of capillaries let us do that. Let us, take the actual red blood cells and the other proteins and stuff that are in our blood that we need to recycle, we need to break down, we need to clean, we need to replace. So these are in the places that do that. The liver and the spleen, those are big ones that are going to be responsible for cleaning up. So cleaning and breaking down red blood cells that are old. And then we think the bone marrow. Within the bone marrow, we have our making new red blood cells. So we have to be able to get those into the bloodstream. So that's through these capillaries. We'll make new red blood cells in the bone marrow, and then we stick them into the capillaries through these sinusoidal capillaries inside of the bone marrow. Most of the bones that make our marrow are the big long bones as well as the pelvis. So mostly think pelvis, think femur, a little bit in the other long bones, but the big long bones where we mostly do that. Finally, the lymphoid organs, all of our lymph stuff that we're going on with. Remember that if we think about the whole lymphatic system plays a couple different roles. Part of its role is development and using a lot of our white blood cell stuff, our immune system cells. So it's helping create our immune system cells. And so it needs to have a way to be able to get those into the bloodstream. And so you have the sinusoidal capillaries. So where the lymph tissue is, it has the sinusoidal capillaries to be able to put in the white blood cells that needs to be put into the bloodstream. As well as think back to when we talked about in biochemistry, talking about the fats and the chylomicrons, all that kind of stuff. Sorry, give me one second. All the chylomicrons and stuff, we need to be able to get those from the lymph tissue into the bloodstream. That happens through the lymph, get to those lymphoid organs where those chylomicrons can transfer back over. As well as the lymph tissue, all of the lymph fluid has its own system. I'm assuming that we'll probably cover that later on in here, probably like the last slide. Yeah, I'll look at it a little bit now. We have this whole lymph channels where the lymph is all the extra fluid where it actually carries a lot of the stuff that we are pushing out of the bloodstream. And that goes into all of our different tissues. And we have our separate little lymph vessels that bring the lymph back and bring it all the way back up to the top. And it drains into the subclavian veins. So we'll look at that a little bit later on. Okay. Those are our sinusoidal capillaries. Diffusion of solutes. So we have the arterial end, the venous end, and we have the capillary in the middle. So the important thing to remember is the arterial side, because the arterial side has natural blood pressure, it's high pressure with the blood, and then low blood pressure, let's just say BP. High blood pressure on the arterial side comparatively, higher blood pressure on the arterial side, low blood pressure on the venous side. When we talked about blood pressure, we referred to blood pressure being the pressure that the blood is pushing against the cells, against the cell wall, or sorry, not cell wall, against the walls of the vessels. So the amount of pressure that the blood is exerting and pushing onto the cell walls. So if you think about it, we're putting pressure against the cell walls. And with the capillaries, we have these little openings. So even our continuous capillaries, we have these little openings. If we're pushing pressure from the inside towards the outside, essentially think of this like a tube of toothpaste or a tube of jelly or something. Or if you've ever, as kids, did you ever play with, they're like the little tube-like things, but they kind of like, it like folds into itself and has like jelly or something on the inside.
Yeah, I used to play with those all the time.
These little things. I kind of imagine the vessels like one of these things, but if you were to poke a bunch of holes into it, so you poke a bunch of holes into it and then squeeze one end, the liquid and the fluid is going to squeeze out. So that's kind of what's going to happen. It's going to part of what's going to be happening when we start looking at like the actual movement of stuff. So let's go ahead and read a little bit of this. So the diffusion of solutes, you remember diffusion. When we talk about diffusion, diffusion is the movement of the stuff that's mixed into the water, typically from an area of high concentration to low concentration. So the solutes will diffuse down their concentration gradient. That is from high concentration to low concentration. Lipid soluble solutes will cross the capillary wall. Stuff like gases, lipid-based hormones, or any other things that are lipid-based. Water-soluble solutes will cross through pores in the capillary wall. Electrolytes and glucose, all that kind of stuff, move with the fluid. Big key point, this next one right here, proteins are prevented from crossing the capillary wall of continuous capillaries due to their size. Notice how it said specifically the continuous capillaries. When we start getting bigger gaps, certain proteins can kind of go through. Do you guys remember what's that main protein that we have in the blood that helps us maintain like our blood pressure and maintain all that kind of stuff? Starts with an A. Good. Yeah, our albumin. Our albumin is going to be one that takes a lot of is going to create and generate a lot of our kind of movement and everything. So we'll come back to albumin in a little bit. The permeability of various capillary beds tremendously differs. For example, it is just like we talked about the sinusoidal versus the fenestrated versus the continuous. We kind of already went over all of that. Depending on how much space there's in between determines how mobile everything is in between it and how much stuff can diffuse and what kind of stuff can diffuse. So you see up in this picture, you kind of have out, in, out, in, out, in, out, in, all this movement. So we'll start to look at this in a second. Understand that there are a couple different forces acting on in this whole system. Okay, we'll talk about a capillary and all of the tissue that surrounds it. We'll consider that our system. Okay, you can think of it a little bit enclosed. That's perfectly fine. So essentially, we learn in physics that whenever you exert a force on something, there's an equal and opposite force that opposes it. Okay, so like we have fluid pressing this way, wanting to get out, but we also have fluid on the outside pressing, wanting to come in. And then we have like our pressures with all of our stuff. Like we have over here, we have a lot of our a lot of albumin, lots of a whole bunch of stuff, lots of fluid and stuff over here, so it's pretty diluted. So we have some of the fluid as of wanting to go out that way, and then here on this other side we have a bunch of stuff mixed into all of this tissue, proteins, whatever. It's going to push the opposite direction. The thing to remember, and we'll look at this a little bit closer, on the arterial side, this is the air. We're doing arrows again, like we did with mean electrical axis. With arrows, we talk about them being vectors. Overall, the direction of force when we add everything up on the arterial end, we're pushing stuff out, because when you add up all of the forces, they go out. And then on the venous end of things, overall stuff wants to come in. And we'll look at exactly how that happens and why. So diffusion across the capillary wall is proportional to the concentration gradient. So basically, the bigger the concentration gradient, the more stuff there is on one side compared to the other, the easier it's going to diffuse. So like O2 and CO2. So because O2 is constantly being consumed, there's almost no oxygen inside of the tissue when the capillary goes through. So if the capillary is going through, and the capillary has a bunch of these blood vessels, it has a bunch of hemoglobin that's holding a bunch of oxygen. And so it's constantly going to be offloading and pushing oxygen across to get into the tissue. Whereas CO2 is being produced by the tissues, so there's a bunch of CO2 constantly in the tissue, and it's going to push it towards, instead push it back the opposite way into the capillary for it to get taken to the lungs and get breathed out. So just saying, the more stuff you have on one side, the more stuff is going to move, basically. This funky word, interstitium. Interstitium is just this free fluid type space in between all of our cells and all of our stuff. It's like we have a cell here, we have a cell here. This is where, you know how there's that joke where it's like, hey, do you, or that pickup line that's like, hey, do you like water? Well, you like 70% of my body then. Ha ha ha. That kind of joke. You guys have heard that pickup line before, right? Hopefully.
Yeah.
Okay. This is kind of where that comes from, where in between all of our cells, there's a little bit of space. And that little bit of space, I should say all of our cells, a good chunk of our cells, kind of like with like the tissue and the fats and everything that's on our structure, there's this space, we call it the interstitium. And within that space, we have free fluid around. We have free fluid vesicles. We have all of this collagen, proteoglycan filaments, all this material whose job is essentially to hold everything together. But within this space as well, we have fluid, and that's where we're going to get transfer of materials. So we look over here, it's 1/6 of our total volume. So 1/6 of our entire bodies is made up of interstitium. So between the cells, and there's interstitial fluid. So this is the fluid. I'm going to write this here. This is where all of that exchange is going to take place. And we got the collagen fibers, proteoglycan filaments. Proteoglycan, that means it has part protein, part carbohydrate, glucose. The proteoglycan's job is to trap water. And then we have reticular fibers that form a mesh-like appearance. Gel, it all kind of has like a gel-like consistency. Diffusion to the gel occurs at 95 to 90%, 99% as rapid as it is compared to free fluid. So like the gel of the interstitial fluid is essentially the same thing as water. So a lot of our old, if we think back to biochemistry in the very first lesson here, when we draw all of our cups with our semi-permeable membranes with water in it and a bunch of stuff mixed in. Essentially our capillary, which is a bunch of fluid, has a semi-permeable membrane, which is going to go into a bunch of other fluid, which is the interstitium, which is that gel type stuff. So that way back in chapter 1, lesson 1, when we did all of this stuff, it's all coming back to that. Oh yeah, woo! It all comes full circle finally. A lot of this that you can read through this, or I'm sure Dr. Tate explained, he might have added little bits to this, but just a lot of this is just the anatomy kind of stuff with everything in it. Edema, when we look at edema, this is when we get the swelling, we get extra fluid into the interstitium and things swell. A lot of that is usually because of issues with lymphatic drainage or things like that. Okay, on to the fun stuff. These are also going to have a handful of AKAs, so we want to start to remember this. So we have what's called capillary pressure, AKA hydrostatic. I'll do this in a sec. We have capillary pressure, which is pushing from the inside of the capillary towards the outside. We have interstitial fluid pressure, which is from the interstitial fluid pushing back towards the capillary. We have plasma colloid osmotic pressure. It's just osmotic pressure, meaning that the pressure is due to proteins and gradients and stuff. I'm going to pause for 1 second. My son is coming to say goodnight. And I'm back. Oh, he's so stinking cute. Okay, where were we? Pressures. Osmotic pressure. So the osmotic pressure is referring to all of the fluid, all of the like the proteins and the stuff that's floating in the water. That's either going to pull fluid towards it or push it away from it. Kind of, for example, we use the example of our little cups again. We have a little free space down here. Kind of like if we have water on both sides. Let's say one side has a ton of stuff and the other side has just a little bit of stuff. There's two ways that this can balance. Either a bunch of this stuff that's in the water can move that way, or fluid from this side can move the other way to balance it out. So we can either this can go to either kind of this situation, where now it's equal concentrations, or it can go and even out itself with the amount of stuff that's in either side. That's what we mean by osmotic pressure. Has to do with all the stuff that's in there and the way that the fluids want to move. I like to think of it like the proteins and all this extra stuff floating around is kind of pulling the fluid towards it. So if we have a higher concentration of something, if we have a higher concentration of stuff in the blood, this side is really clearly concentrated compared to this side. The blood has a whole bunch of stuff pulling fluid this way, and the interstitial fluid just has a little bit of stuff pulling fluid the other way. So in this case, like the blood vessel is going to win when it's pulling the fluid. That's the easy way that I found to describe it. Now, it's important to realize, because sometimes when we draw this, we get it a little bit mixed up with this drawing up here. So it's important to understand at every single point of this vessel, all of these forces are happening. So if we were to take this one single slice right here, so this one little slice right here, and then we ballooned it up really, really big, like zoomed super, super close in, we get this picture right here. This is that teeny tiny slice, that picture right there. So we have this capillary pressure and the interstitial fluid pressure, which are also known as hydrostatic pressures. And we have the osmotic pressure happening at all those points. And we could actually even draw. So if these are our, let's erase all this so it looks clean. So we're getting this capillary pressure. If this is that blue arrow right here, blue here, this is happening at every single point along the vessel, right? Just depending on what part of the vessel that we're looking at, whether this is the arterial side or the venous side, is going to determine which one is bigger. Like, are we pushing more fluid out or are we pulling more fluid in? So now the AKAs, I should ask, any questions or confusion about anything up to this point? Hopefully it hasn't been super confusing.
Very understandable.
Okay. If you have any questions, let me know. For those watching the recording afterwards, if you have questions, leave comments, ask your questions, because I get all the comments get emailed to me, and I see them and I do respond to them if people have questions. Okay. So the AKAs of what you're going to see on exams. Hydrostatic pressure, you have it in the plasma, pushing out, and the interstitial fluid. So you can see it you could see it as hydrostatic capillary pressure. You can see it just as PC. You can see it as plasma hydrostatic pressure. Those are the main ones you're going to get. You can add and make up any additional kind of names. If it says the words hydrostatic, capillary, and plasma, if it says any of those words like kind of combined, any of the three combined, at least two of them combined, you know that you're talking about this pressure right here. That capillary pressure that's pushing fluid out of the capillary. With the interstitial fluid, you can have, you can call it the interstitial fluid pressure. You can call it the PIF. You can call it the interstitial hydrostatic pressure. Should I actually add here? Hydrostatic interstitial fluid pressure. Again, if you see hydrostatic, any of them, or this hydrostatic interstitial pressure, all that kind of stuff right there, that's going to be our interstitial pressure. This guy right there. The osmotic pressures, we have plasma colloid osmotic pressure. Sometimes they'll call this capillary osmotic pressure, or they can just call it plasma osmotic pressure, anything like that. As long as it mentions that it's an osmotic pressure and that it's in the capillaries, that's what it's going to be. And the interstitial, same kind of rules apply. If it needs to mention that it's the interstitial fluid and that it's either colloid pressure or it's osmotic pressure in some form, that's what it's going to say. And we have an equation right here. Our net filtration pressure. So net filtration pressure, net means kind of the sum of. So we add them all up and see kind of what it's doing. So that this is going to tell us if fluid is flowing out of the capillary into the interstitial fluid, or if fluid is flowing into the capillary from the interstitial fluid. This is what this equation is going to tell us. So this is saying if it's positive, if after all this math and stuff, it's positive, filtration occurs. So filtration means fluid is leaving the capillary. If it's negative, absorption is occurring. That means fluid is going into the capillary. Okay. All of these forces that determine fluid movement through the capillary membrane. Remember, we talked about the several different types of capillaries, as well as those capillaries themselves are more permeable or less permeable to specific things. Like our continuous capillaries are not going to be permeable to proteins, to red blood cells, but our sinusoidal capillaries are going to be permeable to those things. That's what determines kind of this coefficient. It's like, okay, we look at what's the type of capillary, what's trying to get out of it, and how much stuff is actually going to be able to get out of it at a time. These give us our rough normals. So normally, in a typical scene, on the arterial side, our PCs are high capillary hydrostatic pressure, also known as our plasma hydrostatic pressure. I'm going to write these a couple of times just because it's important to see all the names over and over again. On the arterial side, it's around 30 to 35 millimeters of mercury. On the venous side, it's at 10 to 15, because remember, blood is always going to be flowing from high pressure to low pressure. So we go from high pressure, we keep going to low pressure. And fluid is going to be coming out on the arterial side. Because of the hydrostatic pressure. We get a mean value. They like to have this be a test question, like what's the mean value of hydrostatic pressure? Hydrostatic capillary pressure, 17.3 millimeters of mercury. They like to use that number in test questions, so just make sure you remember that one. I'd remember the 30 as well on the arterial end and the 10 for the venous end. They're just saying the exact same thing. The osmotic pressure. The osmotic pressure on for the plasma, for the capillary, stays pretty much the same, about at 25. Again, this is thanks to proteins like albumin. And that is always a test question. Always, always, always a test question. What's the protein, or what's responsible for maintaining the osmotic pressure going into the capillary or inside of the capillary, or maintaining the plasma osmotic pressure? Something like that it's going to ask. And this is our albumin. Interstitial fluid, osmotic pressure. There's not a ton of stuff floating around in the interstitial fluid, so it has a much lower osmotic pressure. It has less pull of bringing stuff out of the capillary. So you see, overall, our hydrostatic pressure likes to push stuff out of the capillary. Our osmotic pressure overall likes to pull stuff into the capillary. Here is our equation. So this is going to be the trickiest part of doing this equation. And you see how this is written out with all like extra parentheses and stuff. How I want you guys to write these equations is kind of following this general rule. So let's look at let me grab our mock equation. Especially the first times that you do this, and especially because might have to do it in Word. Especially because it's super, super easy to make a mistake doing this addition subtraction. I want you guys to write these out like this when you solve these equations. As soon as my Word opens, which it will get there. So let's look at this. We have maybe this. There we go. Almost there. insert symbol. Oh, right, insert symbol. Not gonna. When you write out these equations, it's going to be, he might have written that out wrong. Actually, write it out like this. Because some of these are going to be one sec, let me pull something up. Something's scratching in the back of my mind where he didn't include parentheses where he's supposed to. Yeah, this is supposed to be like this. This is why parentheses are important. And this is really going to mess with you if you don't know this. Because you're going to plug this into a calculator, it's going to be completely wrong because not having parentheses. This needs parentheses there, there, and there like that. Because if you know math, that minus there in the middle now completely changes this equation. So you need to have the parentheses in there. Actually, hold on. He might have. Did he? Okay, this equation is just wrong. I'm glad that I saw this. These are the correct equations right here. Let me fix this because this is not. We should let him know that this is not right.
Will do. I'm checking ours to see if it's also the same exact.
Yeah, that is not correct. The way that that's written. I think it gets fixed later on. Or maybe it is correct in like a weird roundabout doing a bunch of different numbers way, but the way that I'm looking at it right now, none of the ways that I've done it makes that work out. Yeah. So here's how this is how it should be written. If we just change, okay, so if we add, wait, if we add make this a plus, and then add parentheses here, and then minus this there, unless maybe he's okay. I think I see what he's doing. I think what he's doing, he's kind of skipping ahead because so this direction right here of the capillary pressure is positive. So this is positive right here. This is negative. This is negative. This is positive. Going out is considered positive. Yeah, I guess I see what he's trying to do. Okay. He's kind of skipping ahead, but it doesn't always. Okay. Personally, and the way that I learned it, and the way that I would think is better to do it like this, like I wrote here, with all the parentheses, with the positive, with the negative, and write it out the way that it's supposed to be written out. Because you need to know positives versus negatives in order for, because down here, okay. That. Okay. Sorry, I just confused everybody. Let me. You can do it one of two ways. You can leave it the way that he has it and just plug in the positive version of the numbers that he gives you. So if he gives you 30 and he gives you 5 and he gives you 8 and he gives you 4 or whatever, you can just input those in there. Hopefully he doesn't give you like the negative values and stuff. If he's giving you this equation like this, the way that I learned it, we had to input which number was negative, which number was positive, and know kind of what direction each one was going. Doesn't look like he did that. He formulated the equation so that you don't have to worry about the negatives and the positives. So I would just, I guess, depending on how, be careful how he asks the question and make sure that everything is in positive numbers instead of negative numbers, because otherwise that'll get confusing really quick. Sorry, that probably confused. I'll leave a note in the comments about that because that made me second guess a lot of what was going on.
I get what you were saying though, and I get what he's doing now.
Yeah, but then I don't understand why he has a negative value here. Okay, where is he getting 30? I don't know where he's getting these values from. Okay, I'm gonna ignore what he's doing, and I'm just gonna show you how to do it by yourself. Let me pull up a different example. Okay, so these are the equations. This is, let me convert this to so the hydrostatic pressure in the capillary plus, what do you say, minus the hydrostatic pressure of the interstitial fluid minus the colloid osmotic pressure of the capillary minus the colloid osmotic pressure of the interstitial fluid. This is one of the ways that I learned the equation. So if we get an example, they'll tell us that the PCCO, COIF, and PIF, they'll give us most of these values, or if not all of them, all of these values. This one's 35. This one's 26. This is 0. And this is 5. We come in here and we plug in all of these values. Equals 35 minus 0 minus 26 minus 5, 35 minus 21 gives us 14. That is positive 14 if what is it? Net filtration pressure is positive. That means filtration. Let me try his equation and see if I can figure out where he's getting these values from. Yeah, I'm gonna email him as well and see if he can explain that equation to me, because the math is not mathing out an equation. I'm gonna try and stop making it I'm gonna stop trying to make it make sense. So I'm gonna rewind and go over this with all this, okay? There's two ways you can write the equation. Capillary, 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 capillary. Just make it a P, just make a P. Colloid osmotic pressure. Hydrostatic. Okay. I'm going to copy paste this into the chat. I would write down these two equations. You can do it in with either of these equations.
Cool.
Net filtration pressure equals the hydrostatic pressure of the capillary minus the hydrostatic pressure of the interstitial fluid, subtracted from the colloid osmotic pressure of the capillary minus the colloid osmotic pressure of the interstitial fluid, all that stuff in parentheses. This is what it looks like if you make it all pretty, like distribute out the negative and flip stuff around and all that kind of fun stuff. And you can make it look pretty like this as well. So we look at the different pressures. We talked about how on the arterial end, we get a net positive pushing out. On the venous end, we get a net negative bringing fluid back in. So this is on the arterial side. 35, or hydrostatic pressure of the capillary. Hydrostatic pressure of the interstitial fluid. Colloid osmotic pressure of the capillary. Colloid osmotic pressure of the interstitial fluid. It gives us all of the numbers. We plug all of those in using our equations. The 35 minus 0, so the hydrostatic pressures, minus colloid of the blood, minus that, gives us positive 14 millimeters of mercury. And because it's positive, we know that means filtration. So we know that more than likely this is on the arterial side of the blood vessel. We look at the other end. And this is on the venous side of things. So we plug in all of our numbers. The hydrostatic pressure in the capillary has dropped significantly because we're moving towards the venous side, which has lower pressure. So that's 16 minus 0, 26 minus 5, because our colloid osmotic pressures typically stay the same. And we get a negative number, negative 5 millimeters of mercury, and that equals absorption. So fluid is coming back into the cell. And that's really the big math component of it, is understanding, okay, there is stuff going out, stuff coming in, and kind of which end are we at? If the number comes out as positive, we're favoring filtration, which means we're pushing water and pushing stuff out. If we're negative, we're favoring reabsorption. So it's going to be coming back into the capillary. And that's due to the decrease in the hydrostatic pressure in the capillary, because our blood pressure in the capillary decreases the closer we get to the other side. Yeah, so I do not understand his equation at all, how he got what he gets. I'm sure he has an explanation for it. Personally, I think that doing it like this makes sense. I would learn it this way and do it this way, because the other way, it looks like it has him adding a negative from somewhere for some reason, and I wouldn't want to deal with that. So yeah, that's what I
all over again.
What was that?
I said it's mean electrical axis all over again.
Yeah, a little bit. See, Starling's equilibrium. This looks like he just has. Okay. So Starling's equilibrium for capillary exchange. What this is saying is kind of weird how he wrote it this way, but basically what this says is the amount that we filter almost equals the amount we absorb back. So when you look at all of the numbers and everything throughout the entire capillary, our outward forces are 28.3, the inward forces are 28. So we overall have a net filtration of 0.3, which means we have more fluid outside of the blood vessel than what we started with. So we have to have a way to get that fluid back into the system. And we do that through our lymph vessels. This is just the Starling's equilibrium. This is all that's saying. There's an equation there. Not sure if it'll make you use the equation or not. But this is the purpose of the lymph vessels. We have tissue, we have excess fluid in the interstitial fluid. So it's going to take all that fluid and bring it back around and put it back into the subclavian vein. We have the thoracic, there's two ducts. We have the thoracic duct, which is the lower body, left side of the head, left side arm, parts of the chest. It's like all of this, essentially. All of this is the thoracic duct. In DAA, you talked about the lymph vessels briefly. So all of this stuff gets put back into the thoracic duct. And then the right side of the head and neck, the right arm and parts of the right chest, this whole chunk right here is going to be the right thoracic duct. So that doesn't sound right. I'm pretty sure there's another name for that right side. I would just be cautious when you see it. Make sure that the right side says the word right and the left says left. I'm pretty sure there's another name for that. What was that?
I said I also recall there being a different name for that.
Yeah, let me... That's because it's not the right thoracic duct. It's supposed to be the right lymphatic duct.
Yeah. Put up too.
Yeah, it should be right lymphatic. The right lymphatic duct and then the thoracic duct. Yeah, I knew that didn't sound right. Yeah, I'll have to let him know about that as well. Both of them, they return the blood to the junction of the subclavian vein and the internal jugular vein, so up there towards the base of the neck. A lot of this kind of stuff. A lot of this isn't super tested on. The stuff that will be tested on, usually the amount of fluid, like 2 to 3 liters per day. Lymphatic moves. They have one-way valves, just like the veins do. Where so like a lot of like movement and stuff helps with the lymphatic movement. The formation of the lymph. This talks a little bit about the protein stuff. Most of like the protein stuff isn't heavily tested. The things that would be, I'd probably focus on like when the concentrations increase, like with the liver and the intestines. That's what I would focus on. which makes sense. The protein concentration increases with the intestines. If we think about our chylomicrons, or fatty acids, they flow into the lymph. And in the liver, we get glucose and other proteins flowing from the liver. So that's why that kind of stuff is gonna increase. And also the liver is cleaning everything, so because it's cleaning a bunch of stuff, it's gonna have extra proteins in it. Bacteria can enter. There's the part of the fats. More detail in Phys 2. We'll get into even more of that. Rate of the lymph flow, the stuff that affect lymph flow, all of, messing with all of our pressures. So if you understand the direction those pressures are, if you increase or decrease the different pressures, that's when you're going to have increased lymph in the lymphatic system. For example, like a decreased colloid osmotic pressure in the capillaries. So liver is damaged, doesn't produce albumin, leads to decreased colloid osmotic pressure of the capillary. So stuff like that. So any kind of conditions that would, like liver cirrhosis, liver damage, liver failure, that kind of stuff would decrease that pressure. Anything that would mess with those pressures are going to affect how much lymph there is. This is just kind of reading general stuff. Lymph flow, the skeletal contraction, the artery pulsation of arteries, any movement helps lymph flow move better, helps control the total amount of volume. A lot of this is just kind of, hey, read through this. Yeah, know the pressures, kind of its impact on how those changes impact filtration or impact the lymphatic system. Sorry for the big trip over with the equation. Again, like I'm just not sure how he got this equation. I'm sure if we use the equation the way that he meant to, it would work. I've just never seen it used that way before. So, yeah, hopefully that's not too big of a trip up for people. I'll show that other equation one last time. Did I confuse you a little bit too much, JC, or did that kind of my backtracking and redirecting work out in the end?
Oh, I'm good. Yeah, I'm
Okay.
chilling.
Good. Yeah, so I would just write these ones down, use these equations, and we'll call it good. All right, my dude. Do you want to keep going, or do you want to call it a night?
I mean, I'm good right now, I think. I feel like I'm sitting in a good spot, like with this material at least, so.
Good. It looks like he has a ton of practice questions in this next.
Yeah, he did all those today in lecture.
Okay. Yeah, and a lot of this local blood flow stuff, only things to really focus on is like more equations and repeat equations. Make sure you understand those, and we can talk more about like what does what with the dilation and all of that later on. So, with that, I guess I'll let you go chill after a long day.
This is actually a nice week. We only had one test, and it was this morning, so.
Was it biochem?
Micro. Yeah. We have
Biochem is
biochem
next week, right?
Huh? Yeah, biochem's next week.
All right, sweet. All right, my dude. Thanks for stopping by.
Thank you, Drake. I appreciate it. You too.